Hello everyone, I am Danish Shah and you are watching Beyond the Syllabus with Danish Shah, a channel that is dedicated to making topics super simple to understand. This is my first video and today we are going to discuss about the microscopic anatomy of the liver. So without further wasting time, let's get started. Before going into the depth of microscopic anatomy of the liver, first in brief we will discuss about the gross anatomy of the liver. So the liver, it is the largest organ and gland in the body. The liver is divided anteriorly into two lobes. Number one is the right lobe. Number two is the left lobe. And this division is possible with the help of a ligament known as falciform ligament. So, the liver is anteriorly divided into two lobes, the larger right lobe and a smaller left lobe with the help of a ligament known as falciform ligament. The right lobe is almost 4 times larger than the left lobe. In adults, the average weight of the liver is about 1.5 kilograms or 2% of the total body weight. Posteriorly, the liver is divided into two additional lobes. As I mentioned, first we have the right lobe, then we have the left lobe. Now we have two more lobes, posterior side, the caudate lobe and the quadrate lobe. So, the lobe which is present between the ligamentum venosum and the inferior vena cava, that lobe is known as caudate lobe. And the lobe which is present between the gallbladder and the ligamentum teres hepatis, this lobe is known as quadrate lobe. So, regarding the gross anatomy, we discussed about the size of the weight of the liver. We discussed about the anterior lobes of the liver and the posterior two additional lobes of the liver. Now let us dive deep into the microscopic anatomy of the liver. The liver is composed of millions of classical hepatic lobules. So, the structural and functional unit of the liver is classical hepatic lobule. Now, what is a hepatic lobule or classical hepatic lobule? It is a hexagonal structure. Hexagon means which has 6 sides. 1, 2, 3, 4, 5 and 6. So hexagon, it is a hexagonal structure. At the center you can see a central vein. So it has a central vein, and at the peripheries, these are the peripheries. These are the peripheries. It has a structure which is known as portal triad. So, hepatic lobule has a central vein at the center, and at the peripheries it consists of a structure known as portal triad or portal area. The portal triad, tri means 3. The portal triad or portal area, it consists of 3 structures. Number 1, it consists of a branch of portal vein which carries deoxygenated but nutrient rich blood. Number 2, it consists of a branch of hepatic artery which carries oxygenated blood, and then we have a branch of bile duct known as bile ductules through which the bile flows. So the lobule consists of a central vein in the center, and at the periphery or at the corners it consists of portal triads ranging from 3 to 6, and then it consists of cords of hepatocytes. Now what is cords of hepatocytes? As we can see in this image, so this is the central vein, this is the portal triad. Now from the central vein some cells are emerging and they are radiating or moving towards the portal triad. These cells are actually hepatocytes. These cells are actually hepatocytes. So, the hepatocytes run from the central vein towards the periphery or towards the portal triad, and these groups or bunches of cells are known as cords of hepatocytes. That means they radiate from the central vein towards the portal triad. So simply they are hepatocytes. They start from the central vein and they march towards the portal triad or towards the periphery. So up till now we have discussed about that we have a hexagonal structure. This hexagonal structure is known as classical lobule or hepatic lobule. It consists of a central vein at the center, and at the corners or peripheries it consists of portal triad. The portal triad has three structures. Number one, it has a branch of portal vein, it has a branch of hepatic artery, and it has a branch of bile duct known as bile ductule. From the central vein towards the periphery or towards the portal triad, a group of cells, just a minute, portal triad, a group of cells emerge or migrate or radiate from the center towards the portal triad. These cells are known as hepatocytes, and collectively they are known as hepatic plates, hepatic plates or cords of hepatocytes. This is the structure of classical lobule, where we can see a central vein, we can see cords of hepatocytes or hepatic plates, and we see a portal triad. So again, this is a central vein, this is a portal triad which has branch of bile duct, portal vein and hepatic artery. From the central vein towards the portal triad are these cells. These are hepatocytes, and collectively we call them as cords of hepatocytes or hepatic plates, as I mentioned earlier. Cords of hepatocytes or hepatic plates. Now, between these cords of plates or hepatocytes or rows of hepatocytes, we can see, so this is the hepatocyte, this is again cord of hepatocyte. Between these two cords we can see a space, this space is known as hepatic sinusoid. This space is known as hepatic sinusoid, and this space is known as hepatic sinusoid. So hepatic sinusoid is formed by which branch and which branch of artery and vein we will discuss later. But right now the important thing is that between, so if we consider this as the hepatocyte cord and this also a cord of hepatocyte, so this space is what we will call as hepatic sinusoid. Okay, now this hepatic sinusoid, so it's a blood vessel basically. So this hepatic sinusoid, it is a very important area because it is the home for Kupffer cells. Sinusoids, these are the structure, these hepatic sinusoids, they contain Kupffer cells. What are Kupffer cells? They are basically specialized macrophages, and their main function is to phagocytose old RBCs, pathogens, cellular debris, and they also function as antigen presenting cells. Antigen presenting cells means they play an important role in immune defense. in launching an immune response against a foreign pathogen. Now, now we have already discussed the hepatocyte. We have already discussed the sinusoid. So we said the space between hepatocytes, hepatocyte plates is the sinusoid. Now, the space which is present between hepatocyte and sinusoid. Now the space which is present between hepatocyte and sinusoid, that space is known as space of Disse. As we can see in the diagram, this is the hepatocyte. So this is the hepatic sinusoid. So between the hepatocytes and hepatic sinusoids is the space. This space is known as space of Disse. Okay, so this space is space of Disse. Now, this space is known as space of Disse. In this, as I mentioned earlier, in the hepatic sinusoid, there are Kupffer cells. But in the space of Disse, there are these specialized cells. These are known as stellate cells or ito cells. What is the function of these stellate or ito cells? Their main function is to store vitamin A and other fat soluble vitamins like D, E, and K. Though they store fat soluble vitamins like A, D, E, and K. So these stellate cells or ito cells, they store fat soluble vitamins. But in chronic liver injury, for example in chronic hepatitis B, chronic hepatitis C, what happens? These stellate cells, they get activated, and once they get activated, they lose this function of storing fat soluble vitamins like A, D, E, and K. Now what they become? They become myofibroblasts. And these myofibroblasts, they start producing collagen. And once they start producing collagen, this collagen leads to fibrosis and eventually cirrhosis. Because collagen, what collagen does? It distorts or damages the liver architecture. So, it's very important to understand that during chronic liver disease, this space of Disse which contains stellate cell, they play an important role in development of fibrosis and cirrhosis. Now, before going into detail about the blood supply through these hepatic sinusoids, blood supply through the portal vein, and this portal triad, first we should understand that the liver has a dual blood supply. What is the meaning of this word dual blood supply? If we take any organ, for example, if we take kidney, if we take liver, the kidney in the liver, it is supplied by a artery, by a single artery. For example, kidney is supplied by the renal artery, and the blood from the kidney is drained by the renal vein. But in the liver receives blood via the two blood vessels, not by the single blood vessel. It receives blood via the two blood vessels. Number one is the hepatic artery. It receives the blood from the hepatic artery, and also it receives the blood from the portal vein. So that is why we say liver has a dual blood supply, because it receives blood from two blood vessels. Number one is the hepatic artery, number two is the portal vein. So 1 and 2. It receives the blood via the two blood vessels. The hepatic artery, which is a branch of basically a celiac trunk. Celiac trunk itself is a branch of aorta. The aorta itself carries the blood, oxygenated blood from the left ventricle of the heart. So the oxygenated blood to the liver is supplied by the hepatic artery. And the deoxygenated but nutrient-rich blood from the gastrointestinal tract. From the gastrointestinal tract, the nutrient-rich blood, the nutrients which get absorbed into the gastrointestinal tract, especially into the intestines, this nutrient-rich blood is first carried to the liver. via the hepatic portal vein. So the reason being is so that the nutrients get stored in the liver, or if there are any what we call the toxin detoxification function, that's why the blood from the intestine or the gastrointestinal tract is first carried to the hepatic portal vein. So the liver receives oxygen rich blood from the hepatic artery, the liver receives deoxygenated but nutrient rich from the hepatic portal vein. Once the nutrients are absorbed by the liver, are stored by the liver, and the oxygen is consumed by the liver cell, then the deoxygenated blood is drained out from the liver by the single hepatic vein. So liver receives blood from the two major vessels. Number one is the hepatic artery and the portal vein. And the liver, the blood which the deoxygenated blood from the liver is drained by the hepatic vein. Now, so this is the hepatic vein, hepatic portal vein. This is the hepatic artery. So the blue one is the portal vein, the red one is the hepatic artery, and as we can see accompanying them is the common hepatic duct. We will not focus on the hepatic duct right now. First we will focus on the blood circulation. If we see first, as the blood flows, first it goes to these two areas. These are portal triads. That means in the lobule, the blood flows first into the portal triad, into the periphery, then from the portal triads, it's carried to the central vein. So if this is the hepatic lobule, first the blood will reach the portal triad, then from the portal triad it will be carried to the central vein. That's why we say in the lobules blood flows from the periphery towards the center. So blood first enters into the portal triad, from the portal triad, so in the portal triad, then the blood goes into the sinusoids. Basically what happens, the portal triad consists of a branch of portal vein and a hepatic artery. These two blood vessels, we can say, then join together, they form the hepatic sinusoids. Sinusoid is a blood vessel which is present between the two plates of the hepatocytes. From the sinusoids, blood goes into the central vein. From the central vein, blood goes into the central vein, this central vein. Then from the central vein, it goes into the hepatic vein and then into the inferior vena cava, then to the right side of the heart, that is the right atrium and the right ventricle. From there, the blood goes into the lungs via the pulmonary arteries to get oxygenated. So we can say the hepatic sinusoid is the area where the arterial blood, which is carried by a branch of hepatic artery, and a nutrient rich but deoxygenated blood which is carried by a branch of portal vein gets mixed in these hepatic sinusoids. Now, sinusoid, what are these hepatic sinusoids? These are not continuous cells. These cells have fenestrations, or we can say they are discontinuous cells. They have small pores or small holes in them. These sinusoids, they have small pores. Similarly, similar fenestrated blood vessels can be seen in the kidney. Glomeruli. The glomerulus, it has an endothelial layer which has fenestrations. Similarly here we have sinusoids. Sinusoids also have discontinuous epithelial cells, or they just have fenestrations or holes. Why these fenestrations? What happens, since these cells are fenestrated, this allows the plasma, only plasma. That means the plasma which contains nutrients, which contains waste products, which contains enzymes and everything and toxins, this plasma leaks into the space of Disse. So as I already mentioned, if this is a hepatocyte, this is another hepatocyte. The space between the hepatocyte, between two hepatocyte place, is this space is known as what we call this as sinusoid. Now the space that is present between sinusoid and the hepatocyte, this space is known as space of Disse. So as the blood flows through this hepatic sinusoid, since it contains fenestrated cells, the plasma leaks out into the space of Disse. Once the plasma gets leaked out into the space of Disse, the hepatocytes, which have a large number of microvilli, they come in direct contact with the plasma. From this plasma, they absorb they absorb or uptake proteins, toxins, nutrients. That is very important. So the function of hepatocyte is performed here actually in this space. So this is the area where the plasma gets leaked out in the space of Disse, and then the hepatocyte, they do their function. Either they uptake and they do uptake, and they uptake the toxins from this what we call this as plasma, or they release or they uptake the nutrients and proteins from this. This is where the liver performs its function. So as I mentioned earlier, blood flows from the periphery towards the central vein. So as it moves from the periphery towards the central vein, so the areas which are close to the periphery or which are close to the portal triad, they must be getting more oxygen than those which are away from the portal triad. And also they must be getting all other toxic or non-toxic substances more than those which are away from the what we call this as central let me repeat it. The hepatocytes or the blood vessels, the areas, give me a minute. So the cells which are closer to the portal triad, they must be getting more oxygen and more metabolites as well as toxic and non-toxic substance compared to those which are away from the portal triad, because blood flows from the periphery, from the portal triad to the central vein. So that is why the periportal hepatocytes, which are near the portal triad, they entirely rely on aerobic metabolism and are more actively involved in protein synthesis. While as the hepatocytes which are near the central vein, they are exposed to, no, I am not saying that they don't get oxygen. They get oxygen, they get nutrients, but as compared to the hepatocytes which are present near the portal triad, they get more. That's why these central hepatocytes which are near the central vein, they get low concentration of nutrient and oxygen. That's why they are mainly involved in detoxification and glycogen metabolism. So clear? So blood flows from the central vein to the periphery. So blood flows through the hepatic sinusoids. Hepatic sinusoids have fenestrations, so the plasma gets leaked into the space of Disse. It is in this space of Disse the liver performs its functions like protein synthesis, detoxification, glycogen metabolism. It uptakes and releases. The liver uptakes, like liver uptakes this what we call toxins from this plasma, and then releases like proteins and other substances into the plasma. It is the area where liver performs its main function. Now the hepatocytes which are very close to the portal triad, these are known as periportal hepatocytes. They are exposed to comparatively high concentration of oxygen and nutrients than those which are more central or near the central veins. So it's very important. So very important to remember that the central hepatocytes, they are more vulnerable to hypoxia and toxic injury. So if there is any condition like hypoxia and toxic injury like paracetamol induced liver injury, the central hepatocytes get more damaged. But if we have viral hepatitis and autoimmune liver disease, periportal hepatocytes are the one which get more affected. Now, as I mentioned earlier, the hepatocytes. Hepatocyte, what was their, what they have on one side, they have space of Disse. If we look at the hepatocyte, these are known as polarized epithelial cell. Polarized epithelial cell means that they have distinct surfaces with distinct function. One surface has one function, another surface has another function. If we focus on this diagram, if we look at the basolateral surface, the basolateral surface, this basolateral surface, what they do, they are facing towards the space of Disse. They are facing the hepatic sinusoid, they are facing the space of Disse. And their main function is to uptake nutrients, drugs and toxins from the plasma, and the secretion of plasma protein clotting factors into this plasma. But the apical surface, this apical surface, they form the bile canaliculi with the adjacent hepatocyte. That means one surface of the hepatocyte forms what? It faces the space of Disse and does the function of nutrient uptake, drug uptake and toxin uptake from the plasma which is leaked into the space of Disse. And it also secretes or releases plasma proteins and other clotting factors into this plasma. But the other surface, which is known as the apical or canalicular surface, it forms a structure that is known as bile canaliculi with the adjacent hepatocyte. That is why we say hepatocytes are polarized cells. They have distinct, their one surface performs one function, another surface performs another function. Now what is bile canaliculi? As I mentioned earlier in this slide, that bile canaliculi, it is basically a space which is created by the canalicular or apical surface of the hepatocytes. So two adjacent hepatocytes, the apical surface or the canalicular surface of two adjacent hepatocytes, they create a space, they create a structure known as bile canaliculi. This bile canaliculi is the structure where bile is secreted. So bile canaliculi are basically narrow grooves. These are formed between two adjacent hepatocytes. If we measure the length of bile canaliculi in the liver, it is more than 1 kilometer. Bile canaliculi, what are they? They are the smallest branches of the bile connecting system. What is the meaning of that? That means first we will have bile canaliculi. These bile canaliculi will fuse together to form the bile canal of herring. Then they will fuse together to form the bile ductule. Then these bile ductules will merge together to form the hepatic ducts. So the smallest unit of this bile conducting system or biliary tree is the bile canaliculi, which is formed between the two adjacent hepatocytes, which is actually, if more precisely if we want to explain, we can say that it is a groove like structure formed from the apical surface of two adjacent hepatocytes. So initially we start with the bile canaliculi. From these bile canaliculi, they then fuse together to form a larger structure which is known as bile canals of herring. These bile canal of herring, they are composed of cuboidal cells, cuboidal epithelial cells. These are known as cholangiocytes. Then these bile canal of herring, they fuse together to form the bile ductule, which is a part of portal triad. As I mentioned earlier, portal triad contains a branch of what? Bile duct known as bile ductule. Now, bile ductule, so initially we have, wait a minute, we have, so first we had a structure like this. This is a hexagonal structure known as, very good, what? Lobule, classical lobule, classical lobule. It consists of a structure known as central vein. Then at the periphery, it consists of a branch of portal vein, a branch of hepatic artery, a branch of bile duct known as bile ductule. And it consists of a cell group of cells migrating from the center, emerging from the central vein and going towards the periphery. These cells are collectively known as cords of hepatocytes. So between these two cords of hepatocytes, we have hepatic sinusoids. We have hepatic sinusoids. Then between the hepatic sinusoid and the hepatocyte, we have space of Disse. Okay. Then we said first we will have bile ductule. Sorry. We will have first we have bile ductule. Then bile ductule fuses together. Bile ductules merge together. They form canal of Herring. They fuse together to form, very good, bile ductule. Then these bile ductules fuse together to form right hepatic duct, left hepatic duct. These two fuse to form a common hepatic. These two fuse, they form a common hepatic duct. Okay. Common hepatic duct. Then we have cystic duct from the gallbladder. Then it fuses to form the common bile duct. Then the common bile duct opens into the duodenum via the hepatopancreatic ampulla or ampulla of Vater. First we have the bile ductule, canal of Herring. Sorry. This is bile actually canaliculi. Bile canaliculi. Then we have canal of Herring. We have bile ductule. We have right hepatic duct. Then left hepatic duct. They fuse together to form common hepatic duct. As the common hepatic duct descends downwards, it meets the cystic duct from the gallbladder. Then cystic duct and common hepatic duct, they continue as one duct. This is known as common bile duct. This common bile duct then opens into the duodenum and releases bile into the duodenum. Along with the pancreatic duct, they open into the duodenum. Now, this was the structure of the bile canaliculi. So, these canal of Herring, they are composed of cholangiocytes. These are cuboidal epithelial cells. Also, bile ductules are composed of cuboidal or columnar epithelial cells. They are also known as, if they are cuboidal, we call them cuboidal cholangiocyte. If they are columnar, we call them as columnar cholangiocytes. Okay, next. Now, one more important thing related to liver is the regenerative power of the liver. The Almighty has given the liver a power of regeneration. That means when the liver tissue is lost, for example, due to toxic injury or due to surgical resection, that part of the liver then gets regenerated. So, Why this happens? This happens because of the mitosis. So if we have this part of liver removed, the remaining part of the liver undergoes mitosis and regenerates to form a complete liver, and this is known as compensatory hyperplasia. So there is no role of stem cells, it is purely a compensatory hyperplasia. So these remaining cells, they compensate. How they compensate? They undergo mitosis, then they form a complete healthy new liver, new part. This regenerated part, this has similar structure and functional capacity as the original liver. So, because of this regenerative power of liver, the liver transplant is possible nowadays. Living donor liver transplant is possible nowadays. What we do, we take a part of liver and then from the recipient and then donate it to the person who is in need. For example, a person who is having chronic liver disease, end stage liver disease, who is in need of a liver transplant. Now, one last thing about the microscopic anatomy of liver, that liver also contains stem cells. These cells are known as oval cells. Till now we have studied about the Kupffer cells, then ito cells which play a role in liver injury. Now we have oval cells, these are known as liver stem cells as well. We have seen in some conditions like we have seen in fulminant hepatitis or chronic hepatitis, what happens when the hepatocytes, what happens in, there are some conditions where the hepatocyte division is impaired, that means the compensatory hyperplasia is impaired. What happens, these stem cells then contribute to the regeneration power of the liver. So usually, as we have seen, that there is no role of stem cells in the regeneration power of the liver. It is purely a compensatory hyperplasia. That means cells undergo mitosis and then they regenerate the power of the liver. They regenerate that part of the liver. But in some conditions, this division, this mitosis gets impaired. For example, in fulminant hepatic failure or chronic hepatitis, at that time these stem cells or oval cells, they play an important role in the regeneration of the liver. We have how we have found in experimental models, we have seen there these stem cells or oval cells, they play an important role in the regeneration of the liver. They are located in the canal of Herring near the portal triads. They are believed to be bipotent progenitor cells. What is the meaning of bi? Bi means two. That means they can differentiate either into hepatocytes or cholangiocytes. Cholangiocyte means part of bile duct epithelial cells. So liver also has stem cells which are known as oval cells. What they do at a time where the liver is not able to divide or regenerate, at this time in the experimental studies it has been found that they are located in the canal of Herring. They play an important role in the regeneration and they are bipotent. Bipotent means they can either differentiate into hepatocyte or cholangiocytes. So we will summarize what we have learned today. So initially we started with the gross anatomy of the liver, right? The right end, the left lobe, then falciform ligament, then two additional lobes, caudate and quadrate lobe. Then we started with the hepatic lobule, which consists of a central vein, portal triad, and the cords of hepatocytes. Then we discussed about what is a hepatic sinusoid, what is Kupffer cell, what is space of Disse, what is ito cell or stellate cell, and the role of these cells in during chronic liver injury. Then we discussed about the dual blood supply of the liver. We discussed about the flow of blood from the periphery to the central towards the center in the liver. Then we discussed about the periportal hepatocyte. They get oxygen rich and nutrient rich blood as compared to the more central hepatocytes. Then we discussed about central hepatocytes are more vulnerable to hypoxic and toxic injury, while as periportal get more affected in case of viral hepatitis and autoimmune liver disease. Then we discussed about what is bile canaliculi, what the structure of biliary tree, and liver regenerative power of liver, and role of stem cells and oval cells in the regenerative power of the liver. One important thing towards the end, one last thing which I want to discuss is that in blood supply, so blood flows from periphery to center in the hepatocytes, but this bile, it flows from opposite, it flows from center to periphery, center to periphery. So this is the point which I wanted to mention. So thank you for your patient listening. If you have any questions, any doubts, or any further topic suggestions, you can comment. So if you want that I should record videos in purely English or mixed language like Hindi, English, or combination, so please comment, and please let me know if there are any improvements which you want. So thank you so much. Share with your friends and have a nice day.